All right, welcome everyone to MIPIN's August webinar. Today our topic is the crab-apple conundrum, and we have two speakers joining us today, Rebecca Scherr and Emma Farmer, both from the Dawes Arboretum. I'm going to be giving them a proper introduction in just a minute, but I always like to give you a little introduction to the Midwest Invasive Plant Network as well, otherwise known as MIPIN. We've been around since 2006. We work across a 10-state province region with the mission of reducing the impacts of invasive plants across this area. Um, we always like to thank our current sponsors, and that will include the Invasive Species Center of Ontario, Forest Control Vegetation Management, Corteva Agriscience, Markham Holman Inc., Landscape Architects and Contractors in Indiana, and Alpha Nurseries in Michigan. So thanks to all of them. And I also want to extend our gratitude to the USDA Forest Service. We currently have a grant with them titled Prevention and Early Detection of Invasive Plants in High Priority Midwest Forests. So their support is also helping make this webinar possible today. And this is a map of MIPIN's region, so you can see we cover a pretty substantial area. And I always like to just point out a few of the resources that we have available, and this is just scratching the surface, but we do maintain an invasive plant list. So if an invasive plant is listed or regulated anywhere in this area, it's on that list. And we just did some recent updates to it, so I encourage you to check that out. We also maintain a controlled database. So if you're managing invasive plants and you can do a customized search and find out all kinds of different mechanical, chemical, different methods for about 50 different invasive plants. We maintain the Woody Invasives of the Great Lakes Collaborative, woodyinvasives.org, and we have a blog there. And if you haven't seen it, there was a blog piece on the crabapple conundrum. So if you want to read more about it, that's another source for you. And then if you need local support to get more involved with invasive species where you live, you can find a directory for cooperative invasive species management areas where you live, also called cooperative weed management areas. And if you work for a SISMA, we also have resources for you. We have contractor directories, native plant and seed directories, best management practices, etc. So find that at moremipin.org. And to stay connected to us, we have a quarterly newsletter and email listserv, both of which you can access by visiting our homepage. We have a brand new Midwest Events page and calendar. So to make it a little bit easier to find future MIPIN webinars, information on conferences, as well as conferences and webinars offered by other partners in the Midwest region. And I'm realizing there's a typo there. It's obviously mipin.org/Midwest-events. We're pretty active on social media, or you can always just send us an email. Okay, so thanks for everyone for being here today. That's the end of my spiel. But we do rely not only on sponsors to support our organization, but individuals. So if you're able to support us at any amount today, we appreciate it. There's a QR code. I'll also drop a link in the chat. If you want to support us in a way that's a little more fun, we have MIPIN merch. It's getting cooler out. We have hoodies, and we have a few different styles and colors. So kind of a fun way to get the word out about invasive plants, and I think look pretty cool doing it. So I'm going to stop sharing and invite our presenters to share their screen. And while they do that, let me give them a proper introduction. So we have with us today Rebecca Scherr. She is the Plant Collections Assistant at the Dawes Arboretum. She supports the growth and sustainability of the Arboretum's herbarium. She works to strengthen plant records, expand the herbarium collection, and advance research of various specimens. Over the past 2 years, she's played a key role in the Arboretum's invasive crabapple research initiative through careful tracking, documentation, and preservation of DNA samples. Emma Farmer is an accomplished and collaborative conservation leader. She serves as the ecosystems manager at the Dawes Arboretum. She currently leads habitat restoration and management across more than 1,500 acres, building resilient species-rich landscapes by limiting invasive encroachment and promoting native species succession. Emma and her team concentrate their research on invasive species, especially invasive crabapple, creating best management strategies for large-scale restoration and long-term ecological health. With extensive certifications and experience, Emma regularly teaches and trains professionals across the field how to properly treat and manage invasive species. She also directs the Dawes Arboretum's prescribed fire program using fire as a powerful land management tool. And I have had the pleasure of hearing both of these women present before, so you are in for a great presentation. So go ahead and take it away.
Thank you so much, Michelle. I appreciate you having us on today, as well as for the great introduction. We're really appreciative to be able to give this talk with you all today. It's on a topic that we're really passionate about here at Dawes. So without further ado, we're going to get started on the crabapple conundrum. So, as Michelle mentioned, Rebecca and I work at the Dawes Arboretum, which is located in Newark, Ohio. Dawes is about 2,000 acres, and we kind of call ourselves a living plant museum. Our founders, Beeman and Bertie Dawes, established Dawes in 1929, and they were also very passionate about connecting others with nature. And this is still very much reflected at Dawes today, with our mission being enriching lives through the conservation of trees, nature, and history. So here's a little photo just of Dawes from the overview. If you know of Dawes, you've probably seen the hedge letters, which can be seen from the sky, and those were installed by Beeman as a way to kind of landmark us. So across the 400 acres of the Arboretum, we have a variety of places and places that people like to go for viewing purposes, as well as cultivated plant gardens. So here's just a few examples of what those gardens are going to look like. Along with that, we also have over 16,000 labeled plants on grounds, with 4 nationally accredited plant collections through American Public Garden Association's Plant Collection Network. On top of that, we also have over 1,000 acres of natural areas. These include woodlands, wetlands, and prairies. But one common theme that we've seen across all of these areas is going to be the encroachment of a flowering woody species, but it's not the flowering woody species that may come to mind.
So bringing up a topic that a lot of you are probably very familiar with, and that is calorie pear. So what we all know is once a very popular street tree used quite frequently in residential landscaping, started to become present in some areas that we didn't want to be seeing it in, and then creating these woody monocultures. So I bring this up today because we are kind of worried that this is leaning towards happening again. So before we get into that, I want to bring up something called the TENS rule. So this is a framework that basically says if you start with 1,000 introduced species, you then take 10% or just 100 that can escape cultivation. From that 100, take another 10% or just 10 more species that could establish themselves in the landscape. From that 10 that establish, another 10%, and you're left with just one that has the potential to become invasive. So on this scale, when you look at the 25,000 introduced plant species in the US, only about 25 of them have the potential to become invasive. So I don't want to scare anybody that is thinking that, you know, we're trying to stop all introduced species, all cultivation. That's not the case. We're just worried about those few, the 0.1% that do become invasive. And that is what brings us to our topic of the day, which is our crabapples. So crabapples, just like calorie pear, are used highly in cultivation. They're beautiful ornamentals because of their big, bright blossoms in the springtime. But when you find them outside of cultivation, they're not so pretty. So surrounding this vehicle are overgrown crabapples, forming this almost thicket-like impenetrable wall. And not only is it ugly, but it is really, really hard to manage. And I want to bring up that there are many public gardens, many other botanical gardens and arboreta that have cultivated crabapple collections, Dawes being one of them. And we are looking at all crabapples and trying to figure out what it could be that's causing the problem that we've been seeing. But it's not just a problem that we're seeing here at Dawes and the surrounding Licking County area. In fact, There's a ecologist named Julian Campbell who comes from Kentucky who noticed this crabapple growing around his area as well. And he published this article where he's comparing what he's calling the invasive Taringo crabapple to the next Bradford pear in Kentucky. And this picture is just a really good representation of how big of a beast this really is. I mean, if you look at that root structure, it is massive, and all of the sprouts coming off of it just really paints a good picture of this beast that we've been dealing with. Now, let's just talk about apples in general. So the genus Malus encompasses all apples. There are 30 to 55 accepted species, and out of this list, we have this one right here, which is our eating apple. Everything else that is on the list are all considered crab apples. We do have 3 crab apples that are native to the eastern United States, so we might see these in our region here in Ohio and the surrounding states. They are the sweet crab apple, Malus coronaria, the Iowa crab apple, Malus iowensis, and the narrowleaf crab apple, Malus angustifolia. So the one that's going to be most commonly found in Ohio, and for everybody in surrounding states, you can kind of get an idea from these maps what you might be seeing. But for us here in Ohio, we're going to probably be seeing mostly Malus coronaria, or the sweet crabapple, here in Ohio. Malus iowensis is not necessarily native to Ohio, although it is native in that eastern United States region. And then Malus angustifolia was actually extirpated from Ohio and only recently discovered, actually. So crab apples are widely known to hybridize. That is actually how we get a lot of the unique and beautiful cultivars of crab apples that are sold throughout the industry. And what you're seeing here in this picture are 3 different trees that all came from the same mother plant. So as you can tell, they don't look phenotypically the same. And just as they aren't phenotypically the same, they're also not going to be genetically the same on the inside as well. And that is because they have different pollen parents. So as long as there are crab apples in bloom at the same time within pollinating distance of one another, they have the potential to hybridize. And that is all a result from open pollination, that we get these different cultivars. But that also kind of poses a concern for what we've been seeing as this aggressive, what we're calling a non-native hybrid, that's showing some pretty aggressive behavior. So there's a few pictures here so you can kind of see what we're seeing and what I mean by aggressive. So based on several characteristics, which we'll talk about in just a moment, we kind of came up with a suspect, which Julian Campbell already pointed out as being Malus taringo. So the similarities that we're seeing between Malus taringo and the invasive hybrids, mostly our biggest key identifier has been these deeply lobed leaves, which you'll see really late in the season. So probably starting around this time and going into fall, you'll see these deeply cut leaves on the fall branches. Now that's going to be different than the spring leaves, which are typically going to be unlobed on the flowering and fruiting branches. But on the back sides of the leaves, they are typically hairy, at least on the veins. And another good identifier are the fruits. So the fruits are going to be small, and they range in color from yellow to orange to red. And most importantly, they do not hang on to any of their flower parts when they reach maturity. So you won't see any sepals or any of the frilly little bits at the bottom of the fruits. The branches have spines, and the flowers are white, and they start as pink buds. So they open up to white flowers. So those are kind of the biggest factors that we've been considering to make us kind of believe that this crabapple is at least somewhat related to Malus taringo. So we're going to look side by side then at the suspected hybrid and a wild collected Malus taringo from China. So these 2 herbarium vouchers kind of show those characteristics that we talked about. You can see the small fruits. And you can see similarities in those leaves with the lobing and how deeply cut they are. And just for comparison, I'm going to bring up one of those native crab apples, Malus coronaria. So this is the one that I said would be most commonly seen in Ohio. And as you can tell, the fruits are much larger on this crab apple, and the leaves have a distinctly different shape to them. And then Malus iowensis, one of our other crab apples that you might see in Ohio, has these ginormous fruits in comparison to the small fruits of this suspected hybrid. And the leaves are also much different, much more round in this crab apple.
So now we kind of want to talk about why we're concerned. And we want to start that with looking at the distribution of this Malus turingo, or the hybrid that we're seeing. So as you look at this map here, you can see the green dots. Those are areas where we have confirmed specimens seen. And some of the purple dots are indicating where it's suspected, meaning we've kind of like, it's been indicated in literature of something that looks like it. We don't have a confirmed voucher of that species. So why should we be concerned? So we've looked at identification, but now I want to talk about some of the ways that we can combat this. And as you look at these pictures on the screen here, you can kind of start to paint the picture of why we're really concerned. So the picture of the small plant there, this was taken from one of our walking trails here at Dawes, meaning that this was mowed throughout the entirety of a mowing season on probably a weekly basis. And you can see it formed a really, really thick taproot. And it was still alive after multiple seasons of being mowed on a weekly basis. The other picture that we have here, the picture paints a picture, but not to the full extent, because what you can't see is that this is actually a 3-foot tall pile of asphalt. So, and it's still growing there today. I've kind of left it as like, just how long can it live like that? And it's still going strong a year later. I took this picture a year ago. And the next picture, this is another one that we took off of our trail system. This is one that its response to being mowed was to start growing horizontally instead of vertically. And I think the door kind of gives you a frame of reference, but it was over 4 feet long. And so it's almost growing the way it presents out in nature when we're looking at it. It's almost presenting as ground cover, which does not seem possible for a crabapple to be growing as ground cover, but unfortunately, that is exactly what we're seeing. It's been highly versatile and highly adaptable to whatever growing environment that it needs to be able to thrive. So the previous pictures we looked at were all on an annual or a weekly mow routine throughout the season. So now we're going to shift it back a little bit and look at something that's being mowed once a year. This is just a tall grass field that we mow typically late fall once and let it go for the rest of the season. And it looks fairly unassuming until we go ahead and look a little bit closer. So all of the reddish pink that you're seeing in that picture is all hybrid crabapple. And as the picture kind of shows, it does more, it paints a better picture than what I can even speak, but we've seen acres upon acres of this. And as you look at that picture, you can see just the general lack of biodiversity within the picture. You can see a few natives straggling in there, but in general, the percent cover is over 90% crabapple. So we've seen mowed on a weekly basis. We've seen mowed on an annual basis. So what happens when you take away the mow entirely and you let something naturalize, so to speak? We look at this picture here, and again, fairly unassuming from a distance, but we're going to go ahead and take a closer look at this understory. Entirely hybrid crabapple in this understory, and it's very, very dense. It's a tangled, spiny mass of crabapple that you can barely walk through, and you cannot ask your tractor or brush hog to handle this type of infestation. Please ask me how I know, or better yet, you can talk to our mechanic. He was not happy with me on this, but So that kind of leaves people in this mindset of how, if it gets this bad, what can I even do? If it's breaking your equipment. So one thing that we've been doing here at Dawes is looking at large scale woody removal. And we began trialing these in 2023. We were using a fecon or a forestry mulcher. Typically, these are used in power line rights of ways as a way to clear the woody vegetation under the power lines. So we were looking to translate this into large scale restoration. So as we kind of go through these pictures, you'll see how this progressed. And as you see, there's a lot of stubble left after. So we made a lot of passes on this, did a lot of clearing just to try and prevent those hazardous tire popping leftover remnants to this. So this is what it looked like after we did multiple passes on this area and got it down to what we felt was manageable for our equipment. But this is only phase one. Thinking back to those pictures that I showed that tolerate, a weekly mow, we knew immediately that this area would be covered in resprouts. So we began trialing a few different methods to address these resprouts. So in this plot specifically, we broke a 1 acre area into 3 treatment styles. The first plot received a mow only. It did not receive any chemical application. The second plot received a mow and a broadcast spray with glyphosate. And then the third plot received a mow and a select spray with triclopyr. And that one, the third one is similar to like a basal bark application, targeting that type of system. So the following season, we went back and we conducted interrupted belt transect surveys on this plot, and kind of looked at the number of resprouts within each quadrat. So the results of this showed that there was a significant difference between the mow only and the chemically treated plots. This indicates to us that mowing alone is not an effective means of controlling this hybrid crabapple. And even the chemically treated plots, there were still resprouts. So I think the one thing that we've learned as we've progressed through this is that regardless of your treatment method, follow-up applications are going to be extremely important. But in general, if we look at the 2 chemically treated plots, they were pretty similar, with the broadcast treatment showing the best results between the 2. So the plot that we just looked at was about 1 acre, and now we're looking to replicate this on a much larger scale. So at the Arboretum, this is what we call our oak savanna. This is a 13-acre plot that was cleared with a fecon or forestry mulcher in the same way that that last plot was. We broke this area into 10 different treatment sites, and all of those sections were given a random number, which was associated with a chemical application. So currently, we're about halfway through this treatment trial. So we don't have any results to share with you today on this one, but we're looking forward to seeing those results and being able to share those with you all soon. And then looking at a couple different additional treatment trials that we've been conducting at Dawes, we are currently in the process of looking at different types of treatments for individual plants rather than looking at it on a holistic scale. So the 3 types of treatments we're using is going to be a cut stump, a basal bark application, and then a hack and squirt application. So we're going through, we're doing these treatments on 20 species or on 20 individuals within each size class. And then we're going back to see which treatment methods has the highest success rate by looking at overall mortality and the amount of resprouts that we're seeing. So here's just an example of what those 3 methods look like. The one on the left is going to be the hack and squirt. The middle is similar to a basal bark. It's not on a crabapple in that picture, just for reference. And then the right is that cut stump application. So once we complete these trials, we really want to be able to get this out and share it with landowners so that we can prevent a small infestation that you may have in your yard from becoming a large infestation, which we've addressed is very difficult to handle. Okay, so that was a little bit doom and gloom, but I do want to tell you all that there is light at the end of this tunnel. We have seen success with the methods that we have tried. So again, this is a picture here of our oak savanna here at Dawes. And this was it prior to any type of treatment. And then, so this was one plot that we've already completed in that replication study, and that is what it looks like today. So we've been able to start with a monoculture with, like I said, over 90% cover of hybrid crabapple to a diverse prairie habitat. And then here's another example, again, within our oak savanna, where you can see 15-foot tall, dense stand, nothing else survive except this poor lonely oak on the edge, to another diverse area that's supporting other wildlife and our pollinators and things like that. So wrapping that portion up, we've kind of looked at identification. So we have our wanted poster drafted here. We have our picture, we have our environmental impacts established, and we have a genus. So all we're missing is our species, which may seem like a very minute detail, but in reality, what we're looking at is essentially a wanted poster with just a first name. So I'm sorry if any of you are named Joe, but just trying to find Joe in a sea of a million Joes is really tough.
So we also talked about how this crabapple, how crabapples in general reproduce and they hybridize very freely. But we wanted to know specifically if there is another reproductive strategy at play here. And we're interested in something called apomixis. So this is basically an asexual form of reproduction that is done clonally by seed. So this brings up a lot of questions because we're looking at Malus turingo as our top suspect. So is this invasive crabapple just Malus turingo gone wild? Is it a hybrid of Malus turingo and something else? Is it a hybrid that has back-crossed so many times with a parent species that it phonetically looks the same as the parent, but genetically is very different and has then cloned itself to put forward those beneficial genetics that it has? So what we started here at Dawes was collecting herbarium vouchers of every crabapple we have in the collection, as well as some of those wild invasive hybrids around the Arboretum property, around Licking County, and some other outlying areas in Ohio. We collected vouchers both in flower and in fruit. So any of those wild trees, we would like tie a flagging tape or some sort of identifiable ribbon onto the tree so that we can make sure we were going back to the same exact tree to collect both flower and fruiting vouchers. We also collected a whole bunch of fruits from each of these trees as well, and we took them back to our nursery where they were cleaned, the seeds were stratified, and then sowed. And the goal here was to look at what comes up in each pot and how the seedlings are similar and different phenotypically to one another. And especially in comparison to the known crabapple species that we have in the collection versus those wild species found around the area. So with the seedlings, as we kept up to 10 seedlings per parent. So as long as 10 survived, a lot of them had many, many more than 10. Some of them had less than 10 germinate. So that was something that we looked at too, was the germination rate and how aggressive that behavior was. And with these seedlings, we were then able to collect DNA samples, both from the seedlings and the parents. And then these seedlings or these DNA samples were stored in silica gel to be sent off for DNA study. And the Ohio Invasive Plants Council took special interest in this mystery because they cannot assess just an entire genus as being invasive. As Emma pointed out, we need that species in order to get it on their radar, to get it onto the Ohio invasive species list. So they have generously funded some DNA research being done, and we were able to be matched up with the Hudson Alpha Institute for Biotechnology. They are located in Huntsville, Alabama, and we have met with a very cool team of people. So Dr. Alex Harkis, who is the faculty investigator, is overseeing the project. And then Dr. Charity Gokritz is the postdoc researcher on the project. And her research is specifically focused on Malus DNA and reproductive strategies, including apomixis. So it was like the perfect pairing to find this team because they could not be a better fit for what we were interested in. And then there's Samantha Johnson, who is an undergraduate and doing some help assisting in the lab as well, especially with the DNA extraction. So first, their study starts with extracting DNA from the parent plants. And then after it's sequenced, they are going into a few different forms of analysis. So one of those being a comparative study on the relationship between the invasive DNA and the known USDA genotypes. So this will kind of give us an idea of how closely related the invasive crabapples that we're seeing are with known straight species of crabapple. So they're also going to be doing kind of like a family tree analysis to determine if the invasives share a maternal parent. So this kind of gives an idea of the relatedness of those invasives with a known species, but it also gives us an idea of how related the invasives are with each other. And then bringing it back to those seedlings as well, like I said, we have up to 10 seedlings per parent. So DNA from each of those progeny will be extracted and then sequenced. And from there, we're going to be specifically looking at how that DNA compares to the mother tree. So if there is an exact match of the seedling DNA to the parent tree, that is a sign of apomixis. And we're very interested in the rate of apomixis that's happening. So in just this example chart right here, we have 3 seedlings, one being an exact clone of the mother. So that would be 33% apomictic. And we don't care how much apomixis we're seeing. We just want to know if it's present or not, and at what rate, because the higher it is, the more for concern we have. Especially looking across 10 seedlings, if it is present in all 10, that is a clear indicator of a highly aggressive apomictic behavior. So that was a lot of genetic DNA language. So just a quick recap on the main points of this DNA study is there's 3 questions we're hoping to answer. One is, what are the known straight species that the invasive Malus is most genetically similar to? Does the invasive Malus share a maternal parent? And to what extent are the seedlings reproduced clonally? So some of you probably have seen this invasive Malus growing in your properties, the areas that you manage. We would love to have samples if you do find what you can identify as the hybrid invasive crabapple. And there's 2 ways that you can send that to us. You can press a specimen in newspaper and then mail it out to us, or you can seal it in a Ziploc bag with some moist paper towel. As long as you send that pretty quickly, we'll be able to put it into a press and get some DNA off of it as well. But if you recall the map of distribution, how we had some of the surrounding Midwest states of suspected crabapple there, we would love to have a confirmed sample if you are seeing it present in your areas.
Again, thank you all so much for coming to our talk today. This is something that we're really passionate about. We're still learning, as the rest of you are. I know we had heard today that there were some other organizations doing similar work. So we're excited to be able to collaborate, share our experiences, and try and beat this species to the punch before it becomes something similar to a calorie pair. So we're definitely open to questions. So I don't know if Michelle, if you want to kind of coordinate that.
Yeah, thank you so much for a wonderful presentation. We have a few questions and then people can still feel free to add them as we're going along here. Um, we had a couple of quick questions. If we could go back to the slides when you were talking about the chemical treatment that you were doing. Um, we had one question about the percentage of triclopyr that you were using and what you were mixing it with, if you were mixing it with diesel fuel or something else.
That is a great question. Yeah. So I have a whole info sheet about our chemical applications that I'm happy to share out. If we could get that person's email, I'm happy to send that. We do not typically do the diesel, mixing it with diesel. We typically use a carrier oil. But I would have to, I believe, please don't quote me on this. I believe our application was 30% triclopyr on a low volume applicator.
Okay. And then I think we showed the cut stump. You showed the three different application methods, the image of
Yeah.
the there. Okay, yeah. So there was just, I think someone wanted a little more insight on how the cut stump application was
Yeah, so
conducted.
for us, a product that we've found really, really helpful to use, it's called a buckthorn blaster. If you just search that name in Google, it'll pop up. It essentially looks like if you play bingo, the dabbers that you play bingo with, you essentially just coat the exterior of that stump. As you can see, we left the middle or the heartwood empty because it doesn't absorb any herbicide through that portion of it. So targeting the vascular system, which is on the exterior of that plant, that one was with Roundup Custom because it was in a semi-aquatic location. So Roundup Custom, they changed the name to Aquamaster quite recently. It's an aquatic safe herbicide, so we put 100% of that into a buckthorn blaster per what the label recommends, and then apply that with a blue dye so that we can keep track of what stumps were treated.
Okay, great. Yeah, I like buckthorn blasters as well.
They're great. They're so
I got
great.
a couple of comments in the chat about that too. Yeah, they're a great tool. Um, okay, so then we have another question here. Let me see. So this says, I'm curious about the soil conditions. Has there been any investigation into whether the crabapple is reacting to certain nutrient imbalances or conditions? Um, and then there was also a question if it's showing any allelopathy, and if there are any plans to try using burning as a control option. So three-part
Great
question.
question. Yeah, those are great. Um, Interestingly enough, there has not been any soils that we haven't found it growing in. It is very much a generalist. We've seen it growing in closed canopy forests. We've seen it growing in prairie type locations, which is mostly what we had pictured. But we've actually seen it growing right on the edge of a wetland, like with wet feet. So it is very much a generalist in that respect. I don't believe we've seen any allelopathic tendencies, but it's just you could almost say that it looks like it's doing that, but it's hard to tell, and we haven't done any testing to see if it's truly doing that or if it is just it's so dense that nothing else can grow. So we have not done any testing, but they grow in such high density that it could appear that way. But in reality, we think it's just based off of those the closed canopy that it creates, not allowing anything else to grow. Prescribed fire is a great question, and I definitely want to do a research trial on that, but that is not something that we have conducted yet, but that is in the works for next year for us to test.
Okay, that sounds great. Before I go to the next question, did you have your email addresses on one of your slides that we could put up in case anyone wants to get in touch with you, or should they contact the Arboretum if they want to?
We can throw them in the chat right now.
Okay. Otherwise, people can contact me and I can put you guys in, together. whichever way you'd like to proceed. Um, Another question is the density from many individual plants or from resprouting and suckering, or both?
Both. Um, it has been very interesting to look at the growth of all of the different plants. We go to some areas where there are just individual trees that are growing with the same structure as almost a calorie pear, you could say. But then you go to the same location, 2 trees over, and it's coming from a base of like six sprouts. Um, and then even on top of that, thinking of that one picture that I showed of it growing horizontally, we've actually seen it do that. It will have like one stem here grow horizontally through the ground and send up another stalk. So they look like 2 completely different trees, but in reality, they have a connected root structure that also makes treatment very difficult, because if you're not getting that resprout that's 3 feet away, you're not going to kill the plant. So we've seen both.
Okay. In terms of, I know you at one of your slides, you said you were accepting samples. Are there any locations that you were particularly interested in getting samples from?
Yes, I would say there definitely is. I'll run back to that map real quick. Ideally, we're looking at samples that we don't already have representation from. So the green states, we have quite a bit of representation from, especially Ohio. But looking at some of those areas where maybe it doesn't even have a dot currently on the map, we don't even have a suspect in that area. We do feel that it's probably present in a lot more places on this map than what is acknowledged, but until it becomes problematic, it remains fairly unassuming to people, and you can just walk right past it. So really looking at those states that are purple or that don't have a dot at all, we would love to get representation from those states.
Oh, and here's a good question. What do other public gardens think about this plant? Is it a problem at other gardens, or is it specific to Ohio?
I would say it is not specific to Ohio, especially looking back at Julian Campbell's article. He's in Kentucky, and so it's present at least in the northeastern part of the US. But again, I think since most people don't know to recognize it, it's gone unrecognized for quite some time. But I think using this presentation and the outreach of it to gain information from other arboreta and other natural areas, we're going to be able to pinpoint more like the range of its impact.
And Michelle, I know you spoke at the OIPC conference about the pig sip program, and that is also really important for other public gardens and arboreta to communicate on this issue and find out from one another if we are seeing the same thing or not. Our sample from Illinois came from just outside of the Morton Arboretum, and our sample from Massachusetts came from just outside of the Arnold Arboretum. So it is really important for the botanic garden world to keep up to date with each other on this, not hide from it, and share the information that one another has.
Yeah, and I can speak from my own experience of working with botanic gardens across the eastern US, that anecdotally I hear a lot of talk about this problem, but the difficulty in getting a specific species ID is one of the hurdles to getting good reporting into, at least into our system. And I think we had a question here about why EdMaps doesn't show Malus turingo. I'm going to suspect that that's the same issue, is that getting even horticulturists and plant recorders can struggle with identifying some of these hybrids. So it is a challenge.
Yeah, and for Malus turingo specifically too, according to the USDA, Malus turingo is not an aggressive plant at all. And in New York, where they have studied it, it posed actually quite difficult to grow. However, in New Jersey and Maryland, Malus turingo is on their invasive list. So I think the issue with that too, with it just being listed, is that we need to study it. We need to know what it is that we're seeing. And that's why the DNA work is so important.
And even the DNA work that we was funded through OIPC, is really just the tip of the iceberg on what is needed for this project. So being able to get more samples and being able to get additional funding to further this project is really the route that we need to go. And so being here with you all today and being able to talk about it is going to continue to hopefully raise awareness on the issue that we're seeing, and again, head it off before it gets to the calorie pair level.
Okay. I see one more question in the chat, so I'm going to ask you that one, and then I'm going to give everyone about 1 more minute to put a final question in, and then I think we'll kind of wrap things up for today. It said, do we have we need a way to have a plant's DNA analyzed. Is there a commercial option for this yet?
That's a good question. That I don't know. Yeah, I'm not sure. I know they're like Hudson Alpha specifically is looking at crabapple, but it's not like available publicly, I don't believe.
Right.
So I don't think there potentially isn't enough need for it at the moment, but if it continues to be a need, perhaps something will become more readily available.
Okay. Well, I want to thank you both again for being with us and for a great presentation and good discussion here of all these different questions. Reminder to everybody who's listening that Rebecca and Emma's emails are in the chat. There's also a link to the blog piece that the Dawes Arboretum contributed to the Woody Invasives of the Great Lakes Collaborative. You can also link to their website and learn more about their other scientific research happening at the Arboretum. And you can find ways to support MIPIN. So all of that is in the chat. Thank you both so much for your work, for bringing attention to this, and for your time today.
Yeah. Thank you. Thank you so much for having us. It was a great time.
Yeah, and this was recorded. I will get this up on MIPIN's YouTube channel just as soon as possible, and appreciate everyone being here today. Hope you have a great rest of your day.